So what I thought I would do is try to give you an update of what we know about the why the disease presents, how it presents. And my presentation is not meant to tell you, how we deal with the problems that the disease is, but try to answer the question that we get from our families of how all the clinical manifestations that the disease presents come about. So, as you know, this was already mentioned, the disease is multifaceted and it presents with different clinical presentations. It goes from the thrombocytopenia to the eczema to the susceptibility to infections, susceptibility to malignancies, and autoimmunity. For physicians, this presents a puzzle because it's difficult to really put together all these different manifestations that come from different organs and systems to connect them to a single cause. And therefore, it becomes really a question mark still now. What all these things have in common, of course, is that they come from mutations of the Wiskott-Aldrich Syndrome gene, and they come from a deficiency of expression of the protein that is made by that gene, so the Wiskott-Aldrich Syndrome protein, or WASp. So I wanted to give you a little background of what that protein does, and that protein is very important for the correct functioning of the cytoskeleton. The cytoskeleton is that structure that we have in each cells of our organism that functions a little bit like our skeleton. The skeleton is made out of bone and it keeps us standing, it keeps us moving. Each cell has a similar organization and is not made by bone, but is made by proteins based on actin. And WASp is very important for the polymerization of actin, which is this protein here that you see in this picture. These are cells. The blue is the nucleus, and these filaments here are made by this protein that is actin. This actin allows these cells, particular cells, to have that shape, but allows also these cells to change their shape when they need to perform some functions, like in this case, when these cells gets in contact with these cancer cells here, you see the actin gets reorganized and goes to the contact point. And this is a short movie that tells you how these things work. WASp is very important for the correct functioning of this molecule, the RP23 complex. Forget the name. It's just a component that allows the polymerization of actin. If WASp is not around, this component cannot work properly. When WASp arrives into the picture, it takes an actin molecule from the cells and it brings it close to this RP23 complex. At that moment, this complex is active and is able to move and go attach itself to a molecule of actin that is already in the cell. And only at that moment, additional actin can be produced. Without that step, actin will be always in filaments and never in a net shape. And therefore, it's very important that WASp is present and is functioning correctly in order for the cells to have all the possible shapes of this actin and perform all the functions that a cell has to do. And these functions are multiple in all cells of the organism, but especially in the cell's immune system. Here, first you see the actin, the blue one is very important when the cells need to divide, and cell division is very important for immune function, when cells need to touch themselves and communicate to each other. But it's very important, obviously, for the function of the cells against the pathogens. This, for instance, it's called the phagocytic cup, is the structure of the cells that the cells enables when it has to englobe an infection agent. The cells needs to move from A to B to go after these infectious agents, and they need actin to do that. And they need actin also to expel these phylopods here that are able to grab bacteria, like in these pictures. And then there are functions that are even a little more subtle, but all the signaling that goes through the cell's immune system, like the B cells and the NK and the T cells, all are based on actin that allows these communications and this messaging to occur from cellular receptors inside the cells. So if you think now about that, all these mechanisms are affected. They are not eliminated, but they are affected. They are reduced in their ability, in their extent in Wiskott cells. Now you start to understand why is it possible that there are so many different presentations of the disease. Now I'll try to tell you how actin problems can explain the different presentation of the disease. As you know, low platelet numbers and the consequent hemorrhages are one of the most common sign of the disease. And thrombocytopenia is defined as less than 100,000, 150,000, but many of your boys have way below those numbers. And as I said, it is very common because it affects the majority of patients. What you also know is that these patients are smaller than normal, and whether or not they are smaller because they are rendered smaller by the spleen that through which they pass, or if they are made smaller because the production mechanism of the cells are not right. This is still unclear. But what we know is certainly that these platelets, as they go through the spleen, are taken away and destroyed. So the reason why, one of the reasons why there are low platelet counts in Wiskott is really a peripheral destruction by the spleen. But there is also evidence that there is a reduction of platelet production by the bone marrow, and this happens because the megakaryocytes, which are these cells here that in the marrow are the cells that produce the platelets, don't make enough of these platelets. The megakaryocytes, as described in this picture, there are these giant cells that at some point of their differentiation fragment themselves into smaller particles, which are the platelets. This process of fragmentation is dependent on actin, and therefore, from what I told you, you can understand that this process can be altered and affected. So we did a study comparing the platelet, the young platelets, so the platelet productions in patients affected with Wiskott, or compare it to the other most common conditions of thrombocytopenia, so the immune thrombocytopenia. And in this case, we looked at the platelet volume as one of the most common indications of or diagnostic sign of Wiskott, and the number of platelet of young platelets. So in the normal range is indicated here. When we looked at 187 ITP patients, we saw a platelet volume that was normal, not unexpected, and a fractions of small platelets, of young platelets, the platelets that should be reacting to the thrombocytopenia to a value that was in the normal range. When we looked at Wiskott patients, we saw, not unexpectedly, as expected actually, that the platelet volume was lower than normal, and the immature platelet fractions was significantly lower than normal in the ITP patients. And that tells us that these megakaryocytes that are supposed to react to the low platelet counts actually do not do that, and they are deficient in producing new platelets. So there is a platelet production problem that it can be reconducted to an actin deficiency. And there has been a long discussion in the field of whether to try to understand if the platelets not only are reduced in numbers, but are also reduced in function. And that is because some patients that have been splenectomized and they have now normal platelet numbers still continue to have bleeding problems. And the question is not fully resolved yet, but if we look at some of the features of the platelets, like in this case at this number of dense bodies, these black dots here, excuse me, you see that the black dots in Wiskott platelets that you see are smaller than the control platelets, are way reduced, and that therefore can point to a functional problem of these platelets that could be contributing to the bleeding episodes and the bleeding tendency of these patients. So the other feature of the Wiskott patients is their susceptibility to infections of all kinds, viral infections like molluscum in this case, bacterial, like in this case of pneumonia, and viral upper respiratory tract infections are very common. Now, as I listed the most common infections here, and you probably know, you've heard that our B lymphocytes, our T lymphocytes are the ones that really are the cells of the immune system that protect us from infections. What you probably have not heard is that these two groups of lymphocytes divide among them the tasks here, and the B lymphocytes are more responsible for protecting us from bacterial infection, whereas the T lymphocytes are more responsible for protecting us from fungal and viral infection. But as I said, since Wiskott patients have all kinds of infections, we may deduce that both B lymphocytes and T lymphocytes functions are affected. And then there is a third arm or a second arm, major arm of immunity, which is this innate immunity that is less effective than B and T cell lymphocytes, but is able to protect us to some extent from all kinds of infections. So if we just go through this mechanism a little bit very quickly, the innate immunity is formed by all these different white blood cells that we have in the blood. And all these cells play their function by utilizing mechanisms like cell motility and migration, phagocytosis, the immune synapse, the production of chemicals in the cells that kill bacteria, and the ability to kill infected cells. All these mechanisms are dependent on actin and can be measured in Wiskott immune cells to be reduced. So that would tell us that a Wiskott patient with a severe phenotype would have a problem with innate immunity that in itself explains part of the sensibility to infections. Then moving away from the innate immunity, the T and B cell lymphocyte-mediated immunity is part of what is called the adaptive immunity. And as I said, the T lymphocytes are the ones that are more responsible for protecting us from viral and fungal infections. These T cells are produced in the thymus, and they do their functions by making use of the immune synapse, that connection between cells that we have seen before, by proliferate in response to infections, by producing growth factors that enhance the immune response. They are called cytokines. Now, the Wiskott T lymphocytes are very different than normal T lymphocytes. As you see in this picture, there is an electron microscopy picture of a T cell. All these protrusions and microvilli that are typical of a normal T cells are much reduced and not very well developed in a WASp T cells. And that is also due to a problem with actin remodeling and polymerization. And all these defects that I listed here are not there for me to talk to you about it, but to tell you that really all these the T cells from Wiskott patients can be demonstrated to have problems with productions like naive T cells or a specific subset of T cells, the CD8 cells. They are not. They're susceptible to die quickly if you take them away from the bloodstream through a mechanism that is called apoptosis, and a series of important functions like the ability to form immune synapse, to proliferate, or to produce cytokines are affected in these cells. So this explains to us why the T cells of Wiskott patients, even though they are not fully absent like they are in other diseases, are not able to really protect the patients from viral and fungal infections. And as I already told you about the B lymphocytes, the difference between B and T lymphocytes is a little bit in what they do. As I said, the B lymphocytes protect us mainly from bacterial infections, but they are generated in the bone marrow and then migrate to the spleen where they mature, and they are the cells that really make the antibodies. So those specific molecules that protect us from bacterial infections. Also here, if we compare Wiskott B cells to normal B cells, we see a series of deficiencies and defects that characterize the Wiskott B cells. But one thing in particular I wanted to mention, because this is really a matter of black and white, there is a group of B cells called the marginal zone B cells. There are the cells that protect us from pneumococcal infections, if you want, and you know how these patients are susceptible to pneumococcal infections. These cells stay in this zone here. I hope you see that this zone is a little darker than the outside. This zone that is outside of the corona is called the marginal zone B cells. And as you can see in the spleen of a Wiskott patient, this zone is really not developed. And that we still don't understand why these cells do not develop in Wiskott cells. We have a theory that also has to do with actin function. Then we study in mouse model. But these findings really explain to us why the susceptibility to infection, especially to pneumococcal infection, is happening in Wiskott patients. Now, as you all know, autoimmunity is one of those problems that affects, that affects the patients, and the reason why that occurs is we can think that is because the normal mechanisms that protect us from attacking our own body or organs are affected in Wiskott patients, and these mechanisms are mediated by a subgroup of T cells they're called the regulatory T cells that are defective in function in Wiskott patients. And this was one of the earlier findings from the research community when they started looking at of the reasons why 40 to 70% of Wiskott patients present one or more autoimmune phenomenon. So I'm going to not do that because of time. But in addition to the problem of T cells regulatory, so the ability of inhibit autoreactions, many of the Wiskott patients have problems with autoantibody mediated autoimmunity, like cytopenias under the example of autoimmune hemolytic anemia, or IgA nephropathy, so that there are production of autoantibodies or antibodies that create tissue damage. That is probably a complications of the mechanism that maintain B cell tolerance. You should know that during B cell maturation, the cells, the B cells, they originate in the bone marrow, naturally contain B cells that are autoreactive. These cells are gradually eliminated from the bloodstream through a series of checkpoints. And what we think is that the Wiskott patients have failure of maintaining or correctly activated those checkpoints. And this is an example of what that results into. If we look at the presence of autoantibodies in assays like this, where the yellow marks the presence of autoantibodies. Here you see the controls. There are no yellow checks, but Wiskott patients and mild Wiskott patients or XLT patients all present a number of autoantibody production that indicates the susceptibility to autoimmune complications. Now, we don't know a lot of why patients with Wiskott have eczema, but there are some initial studies that implicate the fact that the Wiskott patients may be colonized by different kinds of bacteria compared to patients that do not have this immunodeficiency. And this was a study in which patients that have normal atopic dermatitis were compared to patients that have WASp eczema. I'm sure many of you would like to have this little eczema in your boys, but this was the case in which the eczema was not that severe. By really checking what kind of bacteria live on the skin of these patients, our colleagues at NIH were able to show that the controls, so the non-controls, and the Wiskott patients had different distributions of bacteria. And it's difficult to see here because you see how variable is the situations even in the control. But if you look at least up here, you see some yellow that is not present in the Wiskott patients, and those are a specific type of bacteria, the Serratia, that were only found in Wiskott patients that might indicate that the immune system, the alteration of the immune system in Wiskott patients allows for specific combination of bacteria to live on their skin, and that interaction may result in the eczema. Again, these are preliminary results, but at least it shows the differences, and when you start seeing differences, then you have a pathway to a treatment. As you know, also there is this increased risk of malignancies, especially lymphoma in Wiskott patients. And we don't know much about it. You can certainly attribute the risk of malignancy to the immune deficiency. The immune system is there to protect us from infections, but also from cancer. And so we can really explain that simply by taking that into consideration. There are some initial studies that now show that one of the functions of the Wiskott-Aldrich syndrome that it doesn't have to do with actin is in the nucleus, and in the nucleus the Wiskott protein is able to maintain the stability of the DNA, and therefore reducing the possibility of mutations happening. And as you know, mutations in the DNA may cause cancer. This is a figure where you see that the presence of these green dots, which are the abnormal DNA, and the red dots that are a indication of DNA damage, are more frequent in cells that come from mild Wiskott and Wiskott patients compared to control, and that has been interpreted very recently as a susceptibility or a potential explication for susceptibility to cancer in Wiskott patients. And I'll stop there. Hopefully I'm not too late. questions if you want now or later after the next presentation.
I just had a question about the autoimmune slide where you had the graph. Were you comparing? Yeah, graph. Were you comparing? You were saying XLT and plastic.
Yes.
Are there several different slides we're looking at there?
Several.
Several different.
Oh, each of these is one patient.
Oh, okay.
And these are so the Wiskott, classic Wiskott, and patients have been called XLT.
Okay, so XLT over on the right.
Yes.
And then plastic
Yeah.
in the middle.
As you see, there is no difference.
Right.
And that's one of the puzzles that we still have to resolve, whether or not the presence of these autoantibodies is telling us that these patients are going to develop autoimmunity, or it's just a biological sign that the B cells are not working properly, but that at some point they will be exhausted and will not cause autoimmunity. This is only following these patients in the future will tell us.
And you have the non-affected?
These are the non-affected,
The blue?
yeah,
Okay.
and these are patients with lupus, which are the patients that usually make a lot of antibodies, so we use them as a positive.